Hi, sick friends.
And I'm having PTSD from this.
Yes, I'm just looking at it like no.
Flashbacks.
Yeah, I'm using it to record. Okay. So guys, like I said before, this information is very important, but it is not going to be on this week's quiz, but it could be bonus. So it's still, pay attention. Okay, so we're talking today all about mutations, which are just changes in the DNA of a person, animal, bacteria, whatever it is. So mutation is any change in the order of the bases of DNA. So it's important that it's of DNA because mutations don't happen in RNA. You only can have like a DNA mutation. It's not going to be an RNA mutation. Okay, think back to when we talked about mitosis and meiosis. When do you think mutations happen? What part of the cell cycle? Good. So these happen during interphase. And if you want it to be really specific, what do you think it would be? S. Yeah. S, which is DNA replication. Which is what you guys did when you made your model. So like if you had added in the wrong letter to pair with another one of your letters, that would have been a mutation. And that's what this is. Today, we're going to talk about just point mutations, which a point mutation is a mutation that only affects one base pair. So one like C and G or one A and T. So this is only affects / changes one base pair. Oh, whoa, that was crazy. One base pair. And a base pair is an A and a T or a C and a G. So these are really small mutations. There are other mutations that affect like tons of genes, and those would be things like translocations, where you're taking a piece of a chromosome and you're switching it with a different chromosome. So you have like things really mixed up, or you're deleting like a whole piece of a chromosome. This is a way smaller than that, but it still can have big effects, which we're going to talk about. Okay, the first type of point mutation that we're going to talk about is a substitution, which is exactly what it sounds like it is. It's just like when you have a substitute teacher, it's switching one with another. So this is one base is swapped with another base. And remember in DNA, what are the choices for bases? Good, A, T, C, or G. So this is like, you had something that should have been an A, but now it got changed and it's a T. Or it got changed and it's a G. It's switching it with something it's not supposed to be. So for this, I'm going to do an example before we do some real DNA that actually is words. And they're 3-letter words because why? What do you think? Because how many letters are in a codon? 3. So these represent codons. So this is a codon, this is a codon, this is a codon, and same with these two. All of these are codons. Okay. If there's a mutation, we're going to do a substitution. So we're going to take one letter, one base, and we're going to switch it with a different one. So I'm going to write mine like this. That's going to be the same, not mutated. And then I'm going to mutate this C to an H. And everything else is going to stay the same because this is a point mutation that only affects one base pair. Okay. So this moment right here, this would be the substitution mutation. And see how like the sentence still mostly makes sense, but it has a different meaning now than it had before. But it's still readable, like you can still figure out what it says. So that's a substitution. And now we're going to try it with some real DNA, but I made a mistake. So down here where it says the mutated, erase this last codon and make it the same as what's above it. So this should be TCG. Sorry. Okay, now find the mutation. Where is it? Can you find it? Yeah. Yeah, exactly. So the mutation was a substitution and it was this G got substituted with an A. Okay. So now we're going to do the mRNA, but we're going to do it for the mutated one because that's the new DNA that we ended up with. So just yell it to me, what will be the mRNA? Good, U. AGC. And why is it U? Yeah, because we're changing from DNA to RNA. If this was DNA, this would be T's, but it can't be T's because we're doing RNA, so it has to be U's. Okay. Okay, now take your codon chart that you just used for bingo. Find the amino acids for this. Yep. Yeah. And once you're done, you can check your work with mine on the board. That's what you got? Okay. And remember that these would be linked together now to make a protein. But let's take a moment to figure out what it should have been. So this is our amino acid, our codon that was mutated. So this should have been, what should have been the mRNA? GCG, and the protein or the amino acid, yeah, should have been alanine. Okay. And so this is now, because this valine should have been an alanine, this is a different protein and it's going to fold differently and have a different shape than it would have been if it was an alanine there. And this is actually the exact mutation that causes sickle cell anemia. Have you guys heard of that? So if you haven't, sickle cell anemia is a blood disorder that's genetic, and it makes it so that instead of people's blood cells being donuts, they're shaped like crescent moons, and they can actually like hook onto each other and cause like painful blood clots for those people. And it's only because in their DNA, they should have had a G, but instead they have A. And that's what causes it. So a little change like this can actually make a big difference and a big impact on someone's life. Sickle cell is an example. Cystic fibrosis is also like this. PKU, all those, remember when we did Punnett squares and you guys did that one with the diseases? All of those are caused by little tiny things like this. Yeah. This is different from that, but I will talk about that at the end of this because it's connected. Yeah. Okay, so now let's go over here and talk about like what could result from our substitution. So this is an example of this missense mutation. And what a missense mutation is the wrong amino acid is added to the protein. And that can have small effects or it can have really big effects like it does in sickle cell. There's two other options though. And the first one is that instead of the wrong amino acid being added, a stop codon gets coded for. And a stop codon just tells the protein to like cut it off and it's finished. That's called a nonsense because the protein's gonna not work because it'll be way too small. So this is stop codon is coded for too early. And this is going to make a non-functional protein. And then the last option, if you look at your codon chart, do you see how there's like multiple ways that you can get one amino acid? So you could have a substitution mutation that actually gives you the same result as you had before, and that's called a silent mutation. And if it's silent, it's because you can't see it or hear it. So what this is no change to protein. And so no effect will happen. So actually the most common mutations are silent mutations, and you have probably many at this point, but don't know it and will never know it, and doesn't matter because everything's still doing what it's supposed to be doing, so it's fine. All right, that's substitutions. Now we're going to talk about what happens if you either add in a base or you take out a base. So are you ready? All right, so just scroll back to the side and you're going to see this insertion and deletion. Insertion is inserting. So this is base gets added. Deletion is deleting. So this is base gets removed. And both of these types of mutations are going to lead to something called a frame shift. And we'll talk about what that means. First, we're going to do an example with the fake DNA, the sentence. So I'm going to do a deletion, and I'm going to delete this first E. So what I'm going to be left with is TH, and then I deleted the E. And remember, like when you read mRNA, you have to read it in a group of 3. So what happens is that C becomes the third letter in that codon. And then A, T, A, T, E, T, H, E, R, A, T. So this was deleted. Now, see how this doesn't make any sense now? This is a frame shift because every letter shifted over by 1. And because it shifted over by 1, none of the right things are going to actually be coded for. Yeah, Reese. The last codon just wouldn't code for anything. Yeah. So you would only have an amino acid chain of 4, where you were supposed to have a chain of 5. And probably none of them are going to be what you wanted them to be. Okay, let's try it with a real example. So look at this. This is going to be our normal DNA. What's the mRNA? Good. Okay, go ahead and find your amino acids for this one. And once you have it, you can check it with mine. You get it? Okay. So this is our normal protein, alanine, glycine, tyrosine. And now let's see what happens if we have a mutation. So scroll to the side. First of all, let's identify where was the mutation? Where did it happen? Go ahead, William. Yeah, so it could have been this one or this one. Don't really know. I'm going to say it was this one. So this is insertion right here, because if you compare it to our last one, there's an extra C next to that first C that wasn't there before. That's how you know it's an insertion. Okay. All right, so now let's find our new mRNA. Go for it. Good. 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 Okay, now find your amino acids for our mutation. When you're done, you can check up here. And then go ahead and compare this protein to our original protein. Are any of the codons the same? No. Oh yeah, you're right, but not in the same place. Yeah, and it matters. It matters what place they're in. You're right, Reese, sorry. So they're completely different proteins. They're going to fold completely differently, and they're not going to function the same. And that's going to be a problem probably for a person, plant, animal, bacteria, whatever. So this is what a frame shift is. Frame shift is all of the codons after the mutation are changed. But this is still a point mutation, because why? Do you know? Yeah, because you only actually changed one letter. It just resulted in a change of all the ones after. Also, the only time when it would not change all the other codons is when? Can you think of one? What if you could insert or delete more than one base? Yeah, so except if you insert or delete 3 bases, you're going to have just an extra codon, or you're going to be missing a codon. Extra or missing. I'm going to say amino acid, AA. The last thing I want to talk about before I briefly talk about the COVID vaccine, because it is important for me to mention that, is what causes mutations. So if you're ready, you can scroll down to this blue section right here. And there's really 2 options for what could cause a mutation. The first one is the environment. And if there's something in the environment that causes a mutation, that's called a mutagen. Can you think of some? Radiation. Anything else? Chemicals. Yeah, toxins, from living organisms. For sure. Those are good ones. But they can also happen randomly, just like completely by chance. And they happen randomly all the time. The other thing about mutations is that they can cause disease, which tell me one big disease that we've talked about with mutations. Cancer. Also, genetic disorders. Example is sickle cell. But also cystic fibrosis, PKU, Huntington's disease, any of those. But the other thing about mutations is that they also can be beneficial. And can you think of a reason why they would be beneficial? Yeah. So an example of random mutations, especially being beneficial, is allow organisms to gain new traits that can help them survive. And we'll talk about this more when we get to evolution, but one example of this is, let's say there's like a bunch of rabbits and they live in Siberia and it's very cold and very snowy, okay? And then there's a mutation in one rabbit when it's like developing, like it doesn't mutate during its life, it's like born this way. It's born albino, so it has no color to its skin at all. It can survive better because it lives in a snowy place and it can blend in. So it's less likely to get eaten by like a hawk or a whatever, wolf. And it survives longer, so it reproduces more because it lives longer, so it has more babies. And then there's more albino rabbits because those genes got passed on. That's how evolution works, which we'll talk about when we get there. The other thing that's really beneficial and connected to this is that they allow for genetic variation, which is super important for making sure that like everyone in a population doesn't die from the same disease. And without mutations, it would be a lot harder to actually get genetic variation into the population. And it's like one of the most important things for life, that there's variation and differences. Okay, real quick, let me talk about the COVID vaccine, because the COVID vaccine is an mRNA vaccine, which means that when you get that shot, what you're getting is not the DNA of COVID. So like the DNA of COVID is not like putting itself into your DNA, because you didn't even get the DNA from it. You got the mRNA. So when you got the vaccine, you already got it at this point here. So this is in your cells, and your cells then are going to use your ribosomes to make proteins that are going to be the spike of COVID, just the spike, not the actual virus, not the actual DNA, just one spike. And your immune system is going to recognize that one spike as not supposed to be there, and it's going to create antibodies for that spike. So then when you actually get COVID, your immune system is going to see the spikes and it's going to be like, we know this. And it's going to do the immune response very, very fast. So you probably are never even going to feel sick if it's like the exact COVID that you got the exact vaccine for the spike. Now, viruses mutate and the shapes of their spikes can change a little bit. And that's part of why, like, if you got vaccinated and you still got COVID, like, it happens. It's probably not the exact same variant as whatever you got vaccinated for. But that's how it works. And that's why I think it's important for me to tell you that, like, if you got vaccinated, the mRNA after you get it gets broken down. Remember I told you, like, as soon as the mRNA leaves, it starts getting chopped up. If you got vaccinated like a while ago, the mRNA is long gone. It's gone. But your immune system has memory, and it can remember what that spike looks like when you see it next time. That's how it works. Okay. Yeah. Yeah, so they've asked like how long can your body remember to make the antibodies? And the answer is theoretically forever. Like you will have those memory cells forever, but if the COVID virus mutates enough, then those memory cells aren't gonna like activate when they see the new COVID. And so that's why like every year you have to get a new flu shot because the flu mutates really fast. You still have all the memory from the old flus, but it's mutating really fast. And that's like kind of what's happening with COVID too. But like you'll have like memory of that COVID that you got vaccinated for probably forever. Just might not help. Yeah, good question. So she said, is there a difference in like how long immunity lasts for an mRNA vaccine versus like a protein vaccine? And I think it's still a question. Like it's still very new. Like there are other mRNA vaccines besides COVID, but not very many. And I think there's still like a lot of questions related to like how long is this going to last and like, is it better? So IDK. Okay, guys, last thing we're going to do today is we're going to do a little practice on mutations. I'm going to give you a paper on Schoology is the link that you need to get to it. It says mutation simulation. And this is, oh, if you finish this early, I'm going to put up a quizzes on the board that you can join. You might finish early. You're going to put it in the top bin when you're done. Okay. If you want to work at a lab station, you're invited.
We're very focused.
This is not paper talk.
Wait, look. I think this is paper. This is gonna die. Sorry.